Vuoi presentare tu il collega Nijirak? Sì, sì. Nulla. Allora, presento io il primo che è il professore Nijirak. Nijirak è il più grande esperto, l'ho detto prima, vivente di patologie retiniere familiari, ma della trial che sono oggi, che sono esistiti, di quelli che hanno funzionato, di quelli che non hanno funzionato, da cui siamo imparato tante cose. E nell'ambito della terapia genica oggi ha seguito un nuovo gruppo, una nuova avventura con Splice Bio che riguarda alcune patologie, in particolare la Stargardt, ma sarà Anis a correggerci. Devo dire, per noi è un vero onore, è come avere il Pavarotti delle patologie retiniere familiari della terapia genica con noi. Grazie Anis per essere qua, so che ha cambiato i suoi programmi e questo è ancora un onore e anche un atto di grande amicizia. Grazie Anis.
Thank you very much, Professor Cusimano. It's a great honor to be here. I want to thank yourself, the panel here, and also I'd like to thank you, Professor Cusimano, and all of the organizing committee and co-founders of the Macular and Genoma Foundation. It is a great initiative that's been set up a number of years ago, and it's actually a great pleasure to be back here again. And so it's clearly successful in achieving its aims, and we wish it success for the future as well. What I'd like to talk about today is four key things, four key steps within the next few minutes. I'm going to be talking a little bit about the inherited retinal diseases as a whole. Then really the focus of my presentation is going to be Stargardt's disease. I've been working on this for a number of years now, and this is something that's very personal to me as well. And I'd like to also particularly cover the retinal endpoints related to these kinds of rare diseases. And these endpoints are very important because if you do not use the right endpoints for looking at whether the drug works or is safe or not, then there is no way that you can convince the regulatory bodies like the FDA or European Medicines Agency that the drug works effectively and to get approval, and for that to be effective in the community. So I'd like to focus on that a little bit as well. And then lastly, I want to touch on our latest work that we're doing at Splice Bio, which is looking at a gene therapy, and we're a highly innovative gene therapy, a dual vector gene therapy for Stargardt's disease. And I'll share some early preliminary data which looks very promising for us. So with regards to the inherited retinal diseases, and I'll shorten that for IRDs, we know that actually 1 in 2,000 individuals suffer from a inherited retinal disease. And that is very frequent. And out of that, you can see that retinitis pigmentosa is probably the most frequent. A number of different diseases. Stargardt's is the next most populous one with 12%. Now, what does that mean for us? Okay, what that means for the world is that it encompasses 4 million people who suffer from a rare disease like that, inherited retinal disease. And for Italy specifically, and I hope I have the population correct, but it's 30,000 people who will suffer from inherited retinal disease. So this is a huge number. And therefore, even though we say it's a rare disease, it's actually not that rare. And so that's an important message for everybody out there. And therefore, it's important for us to take that on board. Now, this is a slide which covers a lot of the phenotypic heterogeneity that exists in the inherited retinal disease space. It's a very eloquent diagram on the right-hand side which covers the kinds of photoreceptor cells that could be involved, either rods or cones, and on the y-axis, disease progression. And you'll see at the top there various symptoms that patients often present with in the rare disease space. And it's a very eloquent way of showing how complex many of these diseases are. For example, Stargardt's typically involves cone nodes, but actually can have disease progression going from very early on, the vision could be hardly affected, to very devastating blindness almost, central blindness. And therefore, that's a very classic example of a disease that's very heterogenic. Let me expand on Stargardt's disease itself. It is the most common form of inherited retinal juvenile macular degeneration. We know that between 1 in 8,000 or 1 in 10,000 people are affected worldwide. So it's a very common form of rare disease. We know that there are over 80,000 sufferers in the United States and Europe combined. And that's a huge number when you think about it. And if you think about it, over 99% of that population does not have a treatment available for them. There's only one approved therapy, that's the gene therapy that's available for hopefully curing that disease. And that's Luxturna. And we aim to change that with your help. We know that Stargardt's disease accounts for about 12% of the inherited retinal dystrophy-related blindness as well. And that, again, is a huge number. In terms of the genetics, it's an autosomal recessive disorder, and it's caused by biallelic variants in the ABCA4 gene. And that's the principal gene that's the fault here. Because the ABCA4 gene produces either a faulty or no ABCA4 protein. And that's where the problems arise. And I'll delve into that more in the next slide. Unfortunately, or maybe fortunately, we now know that there are over 1,000 mutation types in the ABCA4 gene. And although that's helpful to know this, it does make life a lot more complex for us, because it means that there could be a whole host of different mutation types that underlie Stargardt's disease. And therefore, one patient with Stargardt's disease may not be the same with another, or with another, or with another. And then that makes finding treatments for Stargardt disease very problematic and very difficult in clinical trials. Inherited macular dystrophy caused by these mutations in the ABCA4 gene are usually located on chromosome 1. And the kind of variants are usually missense variants as the predominant feature. But the underlying message there is that Stargardt disease is the leading cause of central blindness in young adults and children. Now, the gene, the ABCA4 gene, is a large gene. It encompasses about 6.8 kilobases. And why is that important? Well, it's important because when you think about gene therapy, typically we tend to use adenoviral vectors called adeno-associated viruses. Now, unfortunately, these viruses, which are vectors which carry the genetic material into the body, have a cargo capacity load. There is a cargo capacity load issue in that they can only tolerate up to 4.7 kilobases. But I've just told you that the ABCA4 gene is much bigger than that. It's 6.8 kilobases. And that's where the problem arises. Therefore, you cannot use traditional single AAV vector gene therapies for Stargardt's disease. You have to be a little cleverer than that. And we'll show you later on how we, as a company, are approaching this issue in a very innovative way. Now, the protein that gene codes for, the ABCA4 protein, essentially is a flipase. It flips all transretinal from the lumen of the cytosol to the cytosol. And so that's very important, because when there is a fault with that gene and there's a fault in the protein, or if the protein's not even there, then what happens is it causes an accumulation of the old transretinal dimer within the eye. And these are precursors for other, what are known as bisretinoid toxins, which accumulate in the photoreceptors of the eye, and these are toxic to the cells themselves. And therefore, they cause the cells to die and adjacent cells to die as well. And before death, there is dysfunction that happens. And that dysfunction means that the cells don't work properly. And what does that mean for you and me? It means that your vision is affected in one way or another. So this is how this abnormal gene producing abnormal protein affects the vision. And that ultimately leads to cellular death and the central blindness that patients with Stargardt disease suffer. Some of the many clinical manifestations are that there's a gradual loss in central vision. There's impaired color vision sometimes. And not all patients will have all of these symptoms. Some patients will have only some of these symptoms. Others will have many of these symptoms. There's delayed dark adaptation. So patients will complain that they have problems going into dark spaces. Cinemas are a no-go area because it's very difficult for patients to adjust going into the cinema in a dark environment and then coming back out again. There's an accumulation of these yellow fluorescent deposits in the back of the eye, which are toxic materials, which are now actually becoming a useful way for us to follow up the progression of the disease itself. And there's progressive bilateral atrophy of the macula. So there's a death of photoreceptors in the macula. And here you can see that as a dark black area on the black and white picture on the right. Now, unfortunately, the disease is complex, as I mentioned earlier, and there are differences in age of presentation and the severity of the actual presentation. And not all the presentations are the same. And therefore, it's important for us to bear that in mind when we are actually looking at clinical trials associated with Stargardt's disease itself. We know that Stargardt's manifests early in life, and in many patients, it tends to be a rapid progression leading to that central blindness. And of course, we know that unfortunately, at the moment, there are no treatments that are approved available for this disease. Now, there are different ways in which Stargardt's disease presents itself. There is the early onset disease, typically in the less than 10 years of age, very aggressive, very rapid progression of disease, leading to loss of central vision very early on, and quite significant loss of central vision as well. There is the intermediate onset of Stargardt's disease, typically occurring from 11 years of age up to about the mid-40s age group, where there are some specific phenotypes, specific things that you can see at the back of the eye, which are these fluorescent yellow flecks, which actually help to diagnose this. The speed of progression is actually intermediate. It's not as fast as the early onset. However, these patients still have very poor vision, legally blind as well, potentially, and so not a good outcome there either. And then there's a third group. A third group of presentation is the late onset, typically occurring in patients who are greater than 45 years of age, 50 years of age. It spares the fovea, which means that patients typically have relatively good central vision. I say typically because that's not always the case. And I say relatively because relative to normal, it's still poor vision. And therefore, these patients still suffer a lot with their central vision. And it's slowly progressive as well. And the three differentiations here are very difficult to work out. And therefore, with my hat on for running clinical trials, it's very difficult to be able to enroll these three different types of patients in one trial and then be able to get a unified outcome. And these are one of the many challenges that we face in running clinical trials in Stargardt's disease. And that's why there's no treatments available so far, because it has been a very difficult area to explore. But nonetheless, there's a silver lining to this cloud, which is that this is a really exciting time to be working in the Stargardt space, because there's a lot going on in terms of potential therapies that could be approved in the near future for patients with Stargardt's disease. And it's important to note that that won't mean that you would be able to see again if you're a Stargardt patient, but what it would mean is the preliminary step of being able to either delay the progression of the disease or even halt the disease progression. I think a few years down the line, our aspirations may be higher in that we would want to probably reverse the disease if we can, or treat it at an earlier stage in order to prevent damage occurring. And I think that's going to be aspirational for us in the future, but one step at a time. Now, I'd like to cover the retinal endpoints for Stargardt's disease. As I mentioned, it's critical to get the right endpoints for Stargardt, because at the end of the day, if you don't get the right endpoints for the right patients, then unfortunately you will never have an advantage, you will never have the potential of getting any drug approved moving forward. There are an array of different functional endpoints and anatomic or structural endpoints. And I'd like to briefly cover some of these. Visual acuity is probably the most common one that everyone's familiar with. You may have used the chart on the right-hand side. You may have used a variation of that called the Snellen visual acuity chart by your optometrist. But this ETDRS chart, which has 5 letters in all of the lines, is the gold standard for measuring visual acuity in clinical trials. We know that the European Medicines Agency will accept 2 lines of improvement as clinically significant change as a threshold for approvability of drugs. However, the FDA have an approvability threshold which is even higher, which is 3 lines of gain, which means that it's harder to get an approval of the same drug potentially in the States compared to Europe. Okay? So perhaps we have a little bit of an advantage here compared to our American colleagues. Unfortunately, there is some noise associated with this test, and typically it's 1 line of vision, or 0.1 logMAR. And that's the noise that you have to get over before you can show clinical benefit or clinical significant benefit that's there. Now, there are variations of best corrected visual acuity that we tend to use. In Stargardt's disease, using best corrected visual acuity isn't always the best endpoint. And the reason for that is that I mentioned earlier that there is central atrophy going on, death of photoreceptors. And you can get death of photoreceptors very early on in the disease in the central macula, and that can affect your central vision very quickly, very early on. Even though the disease may not have progressed that far, you may have a significant drop in vision already. However, conversely, you might have a patient who has a large area of atrophy, of death of photoreceptor cells in the back of the eye, but not affecting that central vision area, the central fovea. And therefore, that patient could have relatively good vision, preserved vision, but have very advanced disease otherwise, because that Stargardt's disease is affecting the photoreceptors quite significantly. And therefore, the visual acuity is not always the best index for Stargardt's disease. And that's why there's a move now away from best-corrected visual acuity to be able to use as a primary endpoint in clinical trials other parameters. And I'll talk to you more about those in a second. However, we always still do measure best-corrected visual acuity because your vision matters at the end of the day. And therefore, we want to be able to assess that. And ultimately, your vision counts. And that's very important for us as well. Microperimetry is an interesting new endpoint, relatively new endpoint that's coming along. And what that does is it measures the retinal sensitivity of the photoreceptors near the center of the macula. And of course, Stargardt's disease affects the central macular area first, before spreading outwards. And therefore, this is a test that you can use to be able to assess the sensitivity of those photoreceptors near the center of the macula. And this could be an early test of showing dysfunction of the cells before they die. And that's why it could be quite important to be able to run this test, because it could be an early signal that actually you've got damage about to start and you could be losing some of those photoreceptor cells. There are various different instruments that we tend to use. The Maya microperimetry, for those of you who may be used to being in clinical trials, you'll be familiar with this. And it's just one of the instruments that we tend to use. And typically, there are thresholds which are set by the FDA and by the European Medicines Agency of what clinically significant change would be. And typically, you would need to have a change of 7 decibels across 5 retinal loci, which are pre-specified. That's meant to be the threshold for clinical significance. And that's quite a high hurdle bar. So we tend to use microperimetry as an endpoint, but it's more of a supportive endpoint rather than the first primary endpoint of showing whether the drug works or not. It is an important diagnostic tool in Stargardt disease. Like I said, it does measure these abnormalities that are going on. And also the more recent microperimeters do allow you to be able to then change your fixation. Because of course we know that once your central macula goes, your fixation area changes as well. And these microperimeters are able to adapt to that. And these are some typical outputs that we can get, which are helpful for ophthalmologists to assess patients with Stargardt disease using microperimetry and trying to see if there are areas surrounding the dead cells where there are dysfunctional cells, cells that are not functioning well, that could be amenable to treatment. And actually, potentially, amenable for reversing the dysfunction into normal function, potentially, in the future. And here's an example on the left-hand side of the image of the back of the eye, a color fundus photograph of atrophy. And that's not great. And you can see that actually with autofluorescence on the right-hand side, you can measure the area of autofluorescence that could be there. And that provides you with a much better way of looking at, looking at the disease itself. And overlying that area this is what the microperimeter reading gives you, is a series of colored dots. The black dots show the area of dead cells, whereas the lighter colored ones, depending on the color, show you the functional ability, the sensitivity of that retina based on light stimulation that's there. So the more colored and more green dots you have, the better the outcome. Contrast sensitivity is also being used in the past. The Pelley-Robson test is one of those. It measures one specific frequency only. Others do exist which measure other frequencies as well. It is an FDA approvable endpoint. However, there's a lot of noise associated with this test, and therefore, typically, it is difficult to follow patients using contrast sensitivity. Now, functional vision is very important. We've become accustomed to the fact that Luxturna, which is the only gene therapy drug that's approved in the ophthalmic area, in their trials, their phase 3 trials, they used something called the MLMT, the Maze test, in other words, which was a multiluminance mobility course, which allowed them to assess how well the drug worked using a practical functional vision test. It basically was a grid on the floor, which patients had to follow under different light conditions. And so that's what this Maze test is all about. And it's an interesting test, useful for some diseases, but not so helpful for others. And so in the Stargardt's area, unfortunately, we don't tend to use it too much because the central vision has gone, but Stargardt's disease is more of a cone-mediated disease. And this test typically favors more the rod-mediated diseases. It is extremely sensitive, measuring from 1 lux right out to 400 lux as well. And those clinical trials that they had showed this 2 lux improvement in vision as a result of that Luxturna gene therapy treatment, which is for a different disease, of course, which is for a disease called LCA type 2. It wasn't related to Stargardt's. And this data did show that actually there is a maintenance of treatment benefit using that gene therapy. Unfortunately, Luxturna, like I said, is only applicable and approved for LCA2. It does not work in other disease types. And so it's not helpful for patients with Stargardt's. There are other mobility courses that exist, other companies that have developed mobility courses, and all of them veer on the same kind of principle of looking at obstacles in the way under different luminances. One interesting concept is being developed by a company called Nanoscope Therapeutics, which is looking at objects on a table and under different luminances, picking out different objects of different sizes under different luminance levels. That's not been validated yet, but it's interesting, and some trials are already using that as an endpoint to be able to work out whether the drug works or not. And I'd like to also then cover the PROs, as we call it, the patient-reported outcome measures. These are often questionnaires that patients are given to assess whether a drug works or not, or how well they're feeling, what specific aspects of the vision are affected by the drug or the disease. And there's a whole array of different patient-reported outcome measures that do exist. The one that we tend to favor more in the IRD space is the bottom one, the Michigan Retinal Degeneration Questionnaire. That has been specifically validated in inherited retinal disease population. And so it's more specific for diseases such as Stargardt and others in this space. I'd like to cover some of the anatomic endpoints as well. This is a color fundus photograph of a patient with Stargardt disease showing the gray area of atrophy that you can see centrally. However, it's very difficult to measure that. And therefore, you really need to have autofluorescence mapping out that area, dark area, which really allows you to be able to quantify the area more specifically by a reading center where there are expert graders that are looking at this and mapping the area out to be able to quantify the amount of dead cells that are there. And that helps us to be able to then see how the patients are getting on as a result of some treatments over a period of months and years. And we map out the area of dead cells. And the idea is that in Stargardt trials, especially the gene therapy trials, we want to be able to slow the disease progression. And ideally, of course, halt the disease progression fully. And like I said earlier, I think the aspiration is that one day we may be able to even reverse some of those changes in the future. And these disease progressions are very important. For example, the patient that is encapsulated on the left-hand side is 1 year apart. The two images of the same eye, 1 year apart, same vision. However, the area of atrophy has progressed significantly. And therefore, that highlights the fact that vision may not, central vision, best-corrected visual acuity, may not be the ideal endpoint to use in Stargardt clinical trials. However, this atrophic lesion size, as we call it DDAF, under autofluorescence, is an acceptable primary endpoint by the FDA and by the European medicine agencies. So now we have a very good endpoint here, which is objective, which is not reliant on patient assessment itself, but it relies on an image that's taken of the back of the eye and graded by a separate set of graders who are experts at looking at the area of dead cells, the number of dead cells that are there. And that seems to be the way forward for us in Stargardt disease clinical trials. There is some new exciting work that's being done looking at something called the ellipsoid zone. Now, the ellipsoid zone is important because it represents the photoreceptor layer, and it represents photoreceptor viability. And it's an acceptable endpoint by the FDA and EMA as well. And therefore, it's quite an important one. And it could be that the ellipsoid zone changes a little faster than autofluorescence-related atrophy lesion size. And if that's the case, then in clinical trials, at least we can actually assess the viability of a drug much quicker by using ellipsoid zone area. And so many researchers, us included, are looking at this to be able to assess whether this is a viable endpoint to use in future clinical trials. Now, lastly, I'd like to just finish off with covering a little bit about our exciting programs that we've got going on with Splice Bio. This is a picture of Barcelona. And for those of you who are not familiar, the white building in the middle is the Central University area where Splice Bio is based. And on the left-hand side is actually, for those of you who are football fans, or for Americans, soccer fans, that's the Barcelona Football Club training ground. And you can just see the edge of part of the Nou Camp Stadium that's being rebuilt. So our offices that are based in that white building overlook the Nou Camp Stadium. So it's a beautiful city, beautiful area for us to be in. Splice Bio as a company is a relatively young company, seeded in 2020. The technology actually emanates from the US, from Princeton University, from Tom Muir's lab. And it's really related to the engineered INTENE technology that's there. Like I said, we're headquartered in Barcelona, in Spain. We have about 40 employees in total. We raised $40 million in a Series A fundraising 3 years ago, and raised $135 million just a few months ago. Now, you might think that's a lot of money, but unfortunately, in the research and development space, typically a drug would take $1.2 billion from start to finish to be able to get approval. So this is a very expensive business, and having $175 million is just a drop in the ocean, unfortunately. So I'm sure that we will need more money moving forward if the success carries on with this and other gene therapies. Nonetheless, we're focused at being able to look at the technologies. You'll see here the pipeline that exists. Our front-running program is called SB007. No James Bond connotations here. This is a potential treatment for Stargardt's disease. We have 4 other pipeline programs in the inherited retinal disease space. 2 of those are already coming up towards the starting of clinical trials. And we know that this technology that we're working on is also applicable for other diseases in other areas, in the neurospace, in the cardiovascular space, and others as well. So it has broad applicability across the board. I mentioned to you earlier that many of the gene therapies that have been approved have been based out of the AAV gene therapies, and the AAV is the adeno-associated viral vector. Now, unfortunately, all of them have this limitation, a cargo capacity limitation of 4.7 kilobases. And that's just the amount of cargo that they can carry, the amount of DNA that they can carry as part of the gene therapy itself, which makes it very difficult for diseases such as Stargardt disease, CEP290 and many others, Usher syndrome and so on, because those other diseases that I mentioned have large genes that are the cause of the defect. And unfortunately, you can't package them in a traditional AAV. And so the way that SpliceBio have approached this is actually to split the gene in half. And so we use a dual vector system. So instead of having one gene that you inject into the eye, we split the large gene into two, and we actually then put both of those genes into the eye. And the idea is that they come together within the eye to form the full-length gene again. And this has been tried a little bit before, but what we've done is we've really taken it to the next step and to the next level of efficiency. And what we additionally do is we actually attach what are known as inteins to each one of these genes. And these inteins are the magical source that we have, which is proprietary. And what those inteins do is ultimately they fuse the two halves together. They bring the two halves together in the right stage within the eye. They don't bring them prematurely. They don't bring they don't bring the two halves together too late. At the optimum time, they bring those two halves together when you need to bring those two halves together to form the full-length gene within the eye, because that's what you want. Now, the engineered inteins are shown to be much more superior than the natural inteins that some other companies, even in Italy, are using. So we have looked at different inteins here, and our engineered inteins are far superior. We actually have head-to-head data against other inteins also, and our engineered inteins are far superior, more efficient, and easier to work with as well. And so we believe that that's a significant benefit potentially in the future for us. And this is how the technology works. So you have these two halves, the two fragments of the gene that you've split into two, and these are packaged into a liquid which is then mixed together. So as far as the surgeon is concerned, when he or she injects into the eye, they are using just one liquid. But that's composed of two separate halves, which will always stay separate until the right magical moment when those inteins come to life. So they're injected into the eye. They then form the double-stranded DNA with the intine fragments that are there. Here, they're in orange and red, the two halves. They then get transcribed into mRNA, again with the intine parts still attached to them. They come out of the nucleus. They then get translated into protein, still with those two halves there, separate, and with the magical intine parts to them as well. And it's at that stage when the proteins are formed that the inteins are automatically programmed to have an affinity of coming together at that stage. And they do so with high efficiency, bringing together the full-length protein at that stage, and the inteins themselves get excised and are then destroyed within the phagosome system, so that you don't have any remnants or any complications or side effects of that. However, the important thing is that they've brought these two halves of the gene together now to form that full-length protein, which is what you want at the end of the day. And you couldn't have injected the full-length protein at the beginning, because I said to you that this gene is too big. So this is a very elegant and very innovative way of bringing the large gene, splitting it into two, attaching these magical inteins to them, and allowing the coding to occur at the protein level and the protein transplicing, as we call it, to occur at that stage to bring the two halves of the protein together. Previously, dual vectors have been used. However, the dual vectors have come together at the DNA level, and they've produced efficiencies which have been very poor, 15-20% efficiencies, which has meant that you had to use very high doses in order to get any kind of efficiency. And of course, using very high doses in the eye means a lot of side effects and issues, and therefore that wasn't very efficient. What we have here are efficiencies over 95% as a result of using the protein transplicing methodology. And we believe that that could be really transformational moving forward in the gene therapy space, especially treating diseases such as Stargardt's and LCA10 and so on. So where are we in terms of the data? Now, unfortunately, in Stargardt's, you do not have very good animal models that predict Stargardt's disease. So you have to take the next best thing. We have looked at the ABCA4 knockout mice, and we see that actually our drug SP007 has very equivalent levels of ABCA4 protein formation. It also localizes in the right space in the retina, which is also very helpful to see. So it's produced in the right level, it's localized in the right space within the retina, and that's very reassuring. But that's just mouse, okay? What about further downstream in this knockout mouse model? What about the effects of this protein? Does it have a functional effect or not? And we can see that actually efficiencies levels of ABCA4 formation of over 80% can occur with our Site2, which is the one that's been chosen to move forward with. And so we know that we can produce ABCA4 to high levels, and it has an impact on A2E levels. A2E levels are those bisretinoid toxins that we were talking about early on that cause the damage in the eye. And therefore, in the mouse model, we can show that our drug has a really beneficial effect in reducing those toxic levels, toxins that are really harmful potentially to the eye. So what does that mean in terms of other models? Well, we've looked at the ABCA4 knockdown rat model as well. And there again, similar things, similar data. I'm not showing some of the data here because it's repetitive. But what's interesting is that we reduce A2E levels and its precursor, A2GPE levels, by similar, 50-60% levels. And if I was to say to you that previously, much lower levels have been shown in animal models by other drugs. And so this really is transformational as we look at the ability for this protein to be able to reduce the toxic byproducts and toxic metabolites. So what about large-eyed animal models? On the left-hand side is the minipig. We have the concern of here, of course, is that, as I said earlier, we do not have a good animal model in Stargardt's disease. So these are wild-type animals. And therefore, what you can do is show expression of your protein in these different spaces. And we can see that in the minipig on the left-hand side, and also in the non-human primate on the right-hand side. Importantly, in the non-human primate, this is the first time where we now have a macula and a fovea that's present. And we can see in the bright red expression levels of the ABCA4 that's been produced as a result of our gene therapy light up that macular and foveal area. And that's really important because you want your drug to have that benefit at that foveal area, which is where the damage is. So this is very reassuring for us. And the immunofluorescence data at the bottom confirms that there's protein there, not just mRNA. So this is very encouraging. So we have our IND open. We have our approvals in place in various other countries across the globe. And we're now embarking in the clinical development plan, as you can see there. We're running a very large observational study, a natural history of disease study, that's actively recruiting now, called the Polaris study. The reason why the name Polaris was given was that many of you know Polaris is a northern star used by many navigators in the past as a guiding tool, and it's still being used as a guiding star. And so we believe that that natural history of disease study will really be the guiding tool for us, the guidance that we need for across the whole of the clinical development program. Because it will include patients where we will then really evaluate the disease endpoints. We will look at the fast progressors and slow progressors. We'll look at how the patients did before treatment versus after treatment. And so this natural history of disease study is a very critical study for us to enroll in. So it's actively enrolling now. And we've now moved forward into the phase 1/2 gene therapy trial, the first gene therapy trial using a dual vector system in Stargardt's disease. And so that's already started now, and we're already moving forward into that study. This is what the trial looks like. The part A is a dose escalation study, and the part B is a dose expansion study. And we're in the dose escalation study at the moment. There are 3 different doses we'll be looking at, low, medium, and high. And although it's early days, the only thing I can say is that actually the patients that have been treated so far have done relatively well in terms of safety. And we're looking at safety, remember here, predominantly at this stage. And then in the second part, we'll be really looking at efficacy. So it's good news so far, early days, but good news, and we'll keep you posted with regards to any future developments that happen. So I want to summarize now. We've learned today that Stargardt's is the leading cause of central blindness in young adults and children. Unfortunately, there are no approved therapies for Stargardt disease. There's a variety of clinical trial endpoints that exist, both anatomical and functional. However, it seems like the atrophic lesion size, which is measured by autofluorescence, which is the FDA-approved study, and actually it's been used historically for getting other drugs approved as well, seems to be the way forward. We've seen that in terms of our SB007 drug with Splice Bio, that the dual vector delivers high levels of reconstituted full-length ABCA4 protein in both small and large animal models. And we've seen data both in small animal models and in large animal models there, which are very favorable to get the IND approval. And you've seen that we now are running a large observational study called the Polaris study, which is actively recruiting. And we are running the phase 1/2 interventional gene therapy study called the ASTRA study, which is also ongoing. And with that, I'd like to thank you for your attention.
So, Anis, you are impressive as usual. So, year after year, your knowledge is growing. But I'm so thankful for you being here for 3 different reasons. The first reason is that you gave to the auditorium and to the scientists, to the researchers, to the ophthalmologists, a wide-angle view of where you are, of what you are doing, and where we hopefully will be. But more, I thank you because as a clinician, most of the time we were asked by the patients, which are quite nervous because nobody is treating them, giving a solution. Say, well, you are going to we went to the moon, now we are considered to go to Mars, and you did nothing for us. No, we are trying, we do the best, we don't sleep during the night to receive the best possible treatment, making available to you. But now as a patient, you have a rough idea how much difficult it is. And the second thing is that, the third thing is that we tried in the past to work together. And we did not been able to do that for some coincidence. We know, and Benedetto Falsini knows why. I hope that in the future you will reconsider our group, and especially the group that works so well with Daniel Palanker, which is a consortium which is still active between Rome Tor Vergata and the Presidio Britannico for the Hospital San Giovanni Addolorata, where we think that we will have available all the technology, the expertise, the surgical expertise, the clinical expertise, and also the rehabilitation in case that you will take part and have looking for a partner in it. So thank you, Anis, for being here. I really have to applaud for your incredible